Hi everyone, and welcome to the first EMBARK webinar. We have two excellent speakers here today. They are Gerry Wright and Pedro Coelho. But before I hand it over to them, I just want to say a few words about the EMBARK program. So EMBARK is a JPI AMR funded program that started last year, and the goal of this program is to, well, we have essentially three different goals. The first is to establish a baseline for how common antibiotic resistance is in different environments. The second goal is to standardize between different methods for resistance surveillance in the environment. And the third main goal of the program is to identify some high priority targets for monitoring of antimicrobial resistance. And this webinar series is part of our outreach, and we have a website as well, which you can see in the bottom corner here, antimicrobialresistance.eu. And among other things, you can find links to the upcoming webinars in this series. So we have 3 more scheduled before the summer break. That's Heike Schmidt in 3 weeks. And then we had to do some rescheduling due to a medical issue. So we have a new webinar different from the ones that have been announced on the website before, which is Gerry Morris, who will talk about the role of water in transmission of antimicrobial resistance. And then on the last day of June, Kimberly Klein, note that the time is slightly different here, will share her work with us. And then in the fall, we have already 3 scheduled presentations, 2 that have been announced before, and then on October 13, the presentation that should have been on June 9th. And you can register for all of the fall webinars on the website, and registration link, for the spring webinars on the website, and for the fall webinars, there will be links posted online soon. And with that said, I don't want to spend more time on this. So I want to introduce our first speaker. So the format here is going to be that we are going to have a first talk for about 30 minutes, and then a second talk for about 10 minutes or 15 minutes, and then we will handle questions together to both of the speakers and try to have a little bit of a dialogue at the end. And our first speakers, and of course you are very welcome to post questions in the chat, for example. Our first speaker today is Gerry Wright. He's the director of the Michael G. DeGroote Institute of Infectious Disease Research, and he is professor at the Department of Biochemistry and Biomedical Sciences at McMaster University in Canada. And his research concerns the molecular mechanisms of antibiotic resistance, as well as the identification of new antibiotics and new antimicrobial strategies. Gerry is also a great writer, and I encourage you to look up his opinion pieces and his review papers, because they are a great read and they are really fun to read. But without further ado, I will hand over to Gerry, who reportedly has some issues with his webcam, so I think he will present mostly with his cam off. Gerry, please.
Thanks very much, Johan. And yes, I apologize. I'm going to turn my camera off when I give this presentation, because for some reason I'm having technical difficulties today. So I'm not being rude. It's just the way it is. So I'm going to do that now, and I'll start sharing my screen. Okay. So Johan asked me to talk a little bit about how we go about measuring and cataloging antimicrobial resistance in a variety of environments. And so I thought what I would do is give you a flavor of what we've been doing in that context over the past, I guess, about 20 years or so, and how that's evolved and where it's going, and some strengths and weaknesses of a number of different strategies. So the first point here is why would you bother to do this in the first place? And I think I'm extremely passionate about the fact that if we want to really understand antimicrobial resistance, how we're going to manage our existing antibiotics, how do we develop new ones, we really have to have thorough understanding of the mechanisms and the variety and diversity of resistance genes that are out there. So this is, my call would be that going forward, what the whole field should be pushing for is a real effort to do molecular surveillance. Let's find out what genes are out there. Let's find out what the diversity is. And of course, from the perspective that antibiotic resistance is usually dealt with, not for this audience certainly, but for the majority of people who think about antibiotic resistance, or thinking about it almost exclusively in the lens of human disease in the clinic. So their molecular surveillance, I think, is frankly limited to a series of genes that, for the most part, have been relatively well studied. It really, I think, is just the tip of the iceberg, as I hope I'll be able to show you going and in some of the work that I'm going to tell you about today. So my objective here is to maybe plant the seed that what we should be doing over the next decade is working really hard to find out what exactly is out there in terms of resistance, diversity, and genomics. So how do we actually catalog resistance? And so the traditional way is through culture, whether that's coming from an environmental sample, or whether it's coming from a microbiome sample or a disease sample. The traditional way to do this is to culture an organism and explore what its antimicrobial resistance phenotypes are. So, of course, this just is limited here to just whatever is culturable. It can take a while for this to happen, depending on the source of the organisms. If they're rapidly growing gut organisms, for example, this could just be 24 hours. But if they're fastidious environmental bugs, it could take weeks for you to do this. The big advantage is that it's really cheap. This is traditional strategies that have been in use for 150 years. And so we know how to do this pretty well. It also provides us phenotypic information, and even gives you the opportunity to very quickly associate the phenotype with a specific species or genus, because of the long history of being able to identify microbes in this fashion. Some of the disadvantages, of course, is that you're really only limited to what you can see. And so that has resulted in a series of efforts to try and bring this traditional strategy into sort of the modern genomic era. So the next way to do this is to use the molecular biology and actually go to the DNA itself. The advantage here is that now we're no longer limited to culturable organisms. And depending on the technique that you're using, you can get your results back in hours. Or if we're looking at something that's known, if you're looking at something new, then of course it could take a long time. The disadvantage of this, in comparison to just culture, is that oftentimes when we do this, especially in a metagenomic context, we can lose some of that phenotype and species information that we get very easily in culture. So there's a trade-off in doing this. But using genetic approaches, you can do things in several different ways. So, So the next step up from culture in the DNA world is to do functional genomics. That is, isolate DNA, fractionate that DNA in some kind of fashion, whether it's by shearing or by limited restriction enzyme digests, clone those into a susceptible organism, and then select on drug. And that will give you access to the antibiotic resistance elements, depending on the efficiency of your cloning and selection that is inside your sample to begin with. And again, it's agnostic of whether it's a human or environmental sample or animal or whatever. The advantage here is that you can get new resistance genes, and I'll show you an example of that later on. The disadvantage is still a lot of work. I mean, there's you got to get DNA, you got to do all this work and clone it. So this takes, frankly, weeks to be able to do this right. But again, it's a great way to avoid some of the drawbacks of culture, which is that not everything grows well in the lab, and allows you to identify new resistance elements, as does culture, which you'll see in some of the other strategies is a limitation for some of the other approaches. So functional genomics is sort of a marriage between molecular and traditional culture approaches. The other way you can do this is if you know what you're looking for, is you can develop sequence-specific PCR primers to be able to pull out specific genes that you're looking for in a metagenome or in a genome. And I'll show you an example of how we've used this very successfully. It's especially useful if there are regions of resistance genes that are common in most alleles or most variants of those resistance genes. And so if you can design primers to those common areas, you could actually generate a significant amount of information on the diversity of resistance out there within a given family. And I'll show you a couple of examples to hopefully clarify what I mean by that. So again, we've got culture, we've got functional genomics. If you know what you're looking for, you can make sequence-specific PCR primers. The next step from that, of course, is you could just go in and just shotgun sequence everything. Now that the sequencing is so cheap, which at one point was unthinkable, 10, 15 years ago, is routine now. The disadvantage of this, of course, is that you have to sift through a lot of DNA to get to the genes that you're interested in. The other disadvantage is that you really can find things that are likely to be resistance genes. It's very hard to find absolutely brand new things this way, unless you have some kind of clue as to what you're looking for. But it's really just a brute force approach that comes out from the fact that it's so much easier these days to just use next generation sequencing to identify or to rapidly identify what's in there. The advantage here again is that this can be pretty fast. You can turn this around often in 48 hours. And then the final method that I'm going to discuss today is a sort of variation of this, but now using very targeted metagenomic strategies where we use bait sequences linked to an affinity capture bead to be able to go into a metagenome, pull out resistance genes that we're interested in, and then leave all the non-resistance associated elements out. And so you're not sequencing a bunch of genetic information that you don't really care about, and therefore you don't have to actually spend a lot of computer time analyzing. So these are the methods that I think are pretty much in use these days. I'm sure there are variants that I'm not, but this is certainly what we're using in my lab and continue to use it, frankly, all of these methods. And I'll give you some examples here. So what we'd like to be able to do is compare these different methods. They vary in their time and of course their cost. And depending on what it exactly is that you're trying to do, then one method will definitely win out over another. And these things are not necessarily mutually exclusive. So one can anticipate using blended strategies to be able to both go after known genes, for example, and look for new ones in a specific environmental metagenome, for example. So culture. So we've used culture very successfully over the years, and especially in looking to environmental resistance. So as I mentioned before, most of the field, traditionally looking at antibiotic resistance, has focused understandably on pathogenic organisms. And it's really been the last 20 years or so where people have thought to think about the diversity that exists outside of the clinic. And we did a study several years ago that started to look at that, and I think really highlighted, perhaps for one of the first times, just the tremendous diversity that there was out in environments that are non-clinical. So sorry about that. And where we took just simple soil samples, isolated organs. So here we cultured organisms from the soil, screened them against antibiotics, and found resistance to all antibiotic classes that were in our sample, whether they were natural products or semi-synthetic versions thereof, or synthetic versions. It didn't matter, that there was so much genetic capacity to countermand antibiotics in environmental organisms already. And it wasn't, our group was not the only one, but others as well. Gautam Dantas, when he was in George Church's lab, was sort of, and Morton Sumner, same thing, found very similar effects. So there's lots of resistance in the environment that is not traditionally captured in clinical samples. I'll give you another example. This is the Leche Gula Cave in New Mexico, the largest and deepest cave in all of North America. It was discovered in the late 1980s and has been sealed from the surface for about 3 million years. And here with my colleague Hazel Barton, we did a sampling, again, culture-based sampling, where we identified a number of organisms, gram-positives and gram-negatives in this case, did the same kind of study, and we found a whole series of novel, or certainly extremely drug-resistant organisms, not obviously disease-causing. So again, it speaks to the remarkable diversity of resistance in the environment. We picked one of those organisms. This is Andrew Pulowski. He was a graduate student in my lab. Now he's a postdoc in George Church's labs. And he looked at this one organism, Painobacillus LC231, in which he found to be resistant to 26 of 40 antibiotics. And if this was an infectious disease story, this will be called, so-called superbug, a term that I'm uncomfortable with, but nevertheless, with all this resistance, it's a pretty remarkable organism. We sequenced the genome. We used a database that we've been involved with since it was an Excel spreadsheet in my lab 20 years ago, called CARD, the Comprehensive Antibiotic Resistance Database. And we used a tool on there called the Resistance Gene Identifier, which will predict a number of genes from known antimicrobial resistance families. The output that you get from this is either perfect, which means that the genes that you're sequencing is the exact gene that's been deposited in CARD. Strict, which is at a certain hand curated cutoff, whether it's 97% or 88%, depending on the family, that this is a likely going to be a beta lactamase of family X. And then there's something called loose that we use a lot, which is just vague similarity to resistance elements, and you can tweak this analysis to be able to figure out what genes you might have in that in your organism, which is in the case of this Painobacillus species that I mentioned, quite impressive. But when we did that, we got a pretty good coverage of the resistance elements, but it wasn't 100% with what we got. So using this sort of CARD analysis, we were able to identify resistance to a number of genes, and we sort of color graded these as we were really confident, or we call it the twilight zone, sort of that loose interaction. And in so doing, we cloned all these genes and confirmed that they were new elements that are associated with resistance to either beta-glycosides or linazolid or streptogramins or what have you. And then there was a bunch of resistance phenotypes that we could not associate with specific genes. And so there we had to use functional genomics. So I think the key here is that, you know, you can use, especially with environmental samples where we're looking at things that are not probably going to be in most databases simply because we haven't looked enough over the years. As we start to add those genes to various databases, we're going to have to do very careful biochemical studies to confirm that these genes are what they are. And when we can't connect phenotype with genotype, we have to use things like functional genomics. And so, for example, I'll just tell you a story about bacitracin. Bacitracin is a peptide antibiotic that's been around since the late 1930s. And it acts by binding to the undecaprenyl pyrophosphate, which is what you can see here. That is the carrier lipid that brings cell wall biosynthetic components from the inside of the cell to the outside of the cell. This is how you move, you know, lipid 2 across the membrane. Bacitracin wraps itself around the pyrophosphate, as you can see here, in a metal-dependent fashion. There's a key hydrogen bond between an asparagine residue right here. And what we found is or at least what Andrew found, is that when using functional genomics, he found a clone that actually could confer this resistance, found a difference of only one mass unit between the resistant organism and the sensitive organism. And what we found using, again, some biochemistry and some organic chemistry was that there was a new amido hydrolase associated with bacitracin resistance that converts this amide into an aspartic acid. So instead of asparagine, we get aspartic acid. That's a negative charge, and negative charge doesn't work, and that's how you get resistance. So it's an example of using both, you know, sort of genomic strategy and a functional genomic approach, but always backing it up with biochemistry to make sure that you're really getting you're really associating a gene with a function. And that's how we're going to build out the environmental resistance databases of the future. We have to make sure that we're not just guessing based on sequence homology. I'm looking at the time here, so I'm going to skip over this part, but this is just another way that we were in the same organism found new members of the macrolide kinase family, and we're able to do some work on that. You can see that if you want. I wanted to bring out, so that's culture and functional genomics. The targeted PCR approach worked really well for us in looking at degraded samples of DNA. So this is a metagenome of 30,000-year-old permafrost in the north of Canada. We were looking for resistance genes there. And here you're not going to get intact genes, you're going to get gene fragments. And so you can't just make PCR primers of, you know, sort of existing genes that are floating around in various databases, because you can't really pick. So what we did in looking at a number of resistance elements, beta-lactamases and tetracyclines, vancomycin, for example, and many others, which we did alignments of modern genes, identified regions that were common, and made redundant PCR primers, and then went into these ancient metagenomes to find genes. And what we found is, because we did controls, we found genes of extinct megafauna like mammoths, and we also found resistance genes to, as I mentioned, vancomycin, penicillin, and tetracycline. So we couldn't use a functional genomics approach because the gene fragments are simply too small. They tend to be less than 500 base pairs in length. And by using a series of well-spaced genes, we were able to identify the entire vancomycin resistance operon in 30,000-year-old permafrost, which consists of 3 genes. And we were able to actually construct a key gene and show biochemically, again, really important to back this up with biochemistry, that what the resistance element was, and show that it was that it was identical in function, not sequence, of course, to existing circulating resistance genes. But we really often, so that's an example of that, of culture, functional genomics, and targeted PCR. Oftentimes we want to measure resistance in very complex systems, whether it's a gut microbiome or the farm or an environmental sample, water or soil or what have you. And so I'm going to talk to you a little bit. So I mentioned to you in the introduction that you can do this approach by shotgun, or you can also use targeted approaches And I'm going to tell you about a strategy that was developed by Alison Guitar, a PhD candidate in my lab. We just reported it about a year ago, prior to the pandemic. Basically, what we do here is we have, again, you isolate metagenomes that will contain a very small number of resistance genes, shown here in blue. We make probes that have target sequences attached to them. This is RNA. So you want to get an RNA DNA sequence. We go in, capture resistance genes, pull those out using stripped avid encoded magnetic beads. And now what we've got is we can do the sequencing, because now we've enriched. So normally, so if the target gene are blue and the non-target genes are orange, by doing this, we should, if things have worked properly, we will get our probes, or sorry, we'll enrich our sequencing project with resistance genes and not non-interesting genes. So the way that we did this is we actually tiled it. So we made probes that every 80 nucleotides across, and then we would tile them across. So every gene that we selected has a large number of redundant of coverage. So we made about 40,000 probes targeting 2,000 genes that we found that we use in CARD. And we also did shotgun on the same sample. So this is just a gut microbiome sample. This is % on target. So again, on target here means resistance genes that we're looking for. And this is about 0.4% of the sequencing that we did using a shotgun was on target. That was pulling out a resistance gene. So here's the difference. Using the targeted or the Bates approach, we're now well over 50%. So 50% of the reads here are on target. We're actually measuring real resistance. Whereas a simple shotgun method, you're down here in the weeds. So clearly, if you're thinking about cost, time, everything in life that you want hold precious, the Bates approach makes much more sense. The question is, are we missing anything? And so, when we were looking at this single sample, this is the number of genes that we identified that were unique to the Bates approach that we missed using shotgun. And this is these are the genes that we missed using Bates. And clearly looking at these shotgun, genes that we pulled up in shotgun, but were not in our Bates, we just simply didn't have those genes because a lot of them were efflux systems. So I think, the Bates approach for us is the way to go as opposed to shotgun going forward in the future. This is just another example of this. So this is over a number of different samples, over a number of different classes of antibiotics with different mechanisms. Using shotgun, this is the sort of coverage. Dark blue is the darker the blue, the increased number of reads through the sequencing. Again, these are individual samples. So a shotgun is just simply not delivering when it comes to this. So you get a much more thorough gene census using the Bates approach. And you can compare that now. So if you go back to CARD and you start to think about, if I got a gene, if this is a sample, from a wastewater treatment plant, how does this compare? How does this aminoglycoside resistance gene that I'm pulling up compare to what's existing out there in the clinic? Well, if you go to CARD, you can actually, on a very regular basis, go to the NCBI and run the RGI on assembled genomes of known pathogens, and you can pull that together and compare it. And we're doing this now. This is my last slide. We're doing this now with a bunch of samples. This is a study that's ongoing using on the gut microbiomes of neonates, some of which are getting probiotics and some of which are not. And it's really the only way to go forward and get these small samples. So it's really important to measure AMR if you want to understand what genes are present, their diversity, and how they change. All of these methods are useful. Culture is useful. It's really inexpensive. Next generation sequencing is is really great if you know what you're looking for, but you really need to understand what the goals of your study are to make sure that you get the right methods. And I'll just finish by thanking again Allison, who did a lot of the work here, and a lot of its collaboration on the Bates has been with Andrew MacArthur's lab. So I'll finish there. 2 minutes over, and turn it over to Louise.
I just like to say that that's not 2 minutes. 2 minutes over on my watch, so so I think you're fine. Thank you very much for a great talk, Gerry. I am going to hand over to Luis in a second, but I think this is actually providing a very good backdrop to his presentation. I want to remind you again, if you have questions for Gerry or for Luis, please put them in the chat, and we'll take them up at least as many as you have time for in the last 15 minutes of this webinar. So our next speaker is Pedro Coelho. He is the principal investigator of the Big Data Biology Lab at Fudan University in China. His work concerns analysis of microbial communities in different types of environments, from the marine environment to the human gut, and he's using computational methods and metagenomics to address these questions. Luis' lab not only applies these tools, but he also develops new computational methods and to solve these kind of large-scale problems. And I should also mention Luis is one of the 6 PIs that are involved in this EMBARK project that is standing behind this webinar series. So please, Luis.
Thank you. I'm going to assume that you can see my slides and hear me. So, well, a bit of a tough act to follow, but I'll try. So, very briefly give you an introduction to how we are thinking about looking at AMR and quantifying at very large scales, in fact, at the global scale. So we mostly work with metagenomics. So, we start with the knowledge that bacteria, they live their lives in these communities doing their thing, and then we come in and very, very rudely, extract their DNA, and actually we extract a very, very tiny fraction of the overall DNA that's present in the community. We call it high throughput, but it's still a very tiny fraction. And this goes into this sequencing machine, and we get out this metagenome. So this pieces of DNA from all of the bacteria that were there. And so now the question that I've been working on is to use Gerry's expression, how do we sift through all of this data to find to make sense of it? So we have this metagenome, and the first thing we do is we process it by assembly to build these longer pieces of DNA, so-called contigs. And then we run our prediction. So we get computational methods that say, we predict that this little piece of DNA here and there are genes encoding for proteins. And so we do this not just not just for one metagenome, but actually actually we do this for many metagenomes. And because we are looking at the data that's publicly available on resources such as the European Nucleotide Archive. And as we do this multiple times, obviously we are going to be recovering the same sequences multiple times, because we have organisms that are present in multiple samples, and so we get that sequence twice, or we have sequences that travel between organisms, mobile elements. And so we want to get rid of this redundancy while still allowing for a tiny bit of variation. And the approach we chose is to have a species level cutoff. So 95% nucleotide identity, which, albeit not perfect, roughly corresponds to the diversity that's present between the strains of a single species. So we do this, and then it's these so-called unigenes that then we analyze. So for example, we run RGI that was just mentioned to find the antibiotic resistance genes, which we can then link back to the metagenomes from whence they came, to try to make sense now biologically what's going on. So as I said, this is an ongoing project, and we've built this, what we're calling the GMGC, the Global Microbial Gene Catalog. And in this first version, so we've taken just over 13,000 metagenomes from a variety of habitats, so including human-associated habitats, the human gut and others, but as well as sort of environmental, marine soil, etc. And we've also added all of the open reading frames from the Progenomes2 database, which is a database of isolate genomes that's maintained by the, or started by the EMBARK lab, and it's now maintained by Daniel Menda. And we start with several billion of these open reading frames, and then we process them into these species level unigenes, as well as broader homologous families. So just to give you a little bit of a couple of numbers behind this. So most of the samples are from the human gut, because this is what the field has done and what's most available. But we've made an effort to look at other animal associated microbiota, as well as the environmental samples. And all of these data were reworked from scratch and using a consistent methodology. So at least from the computational point of view, all of the data was processed similarly. And part of the reason is that one of the driving biological questions was, what kind of sequences do we see show up again and again across environments? And perhaps not so surprisingly, we don't see a lot of sharing. So most genes, and so in this plot here, so the width of these ribbons represents how much sharing is going on, and there's slightly different ways of quantifying this, and you can ask at different levels, but we get very, very, very consistent answers that there's not a lot of sharing. So most genes, most sequences are habitat specific, perhaps with the exception of and I'm going to zoom here on the right side of the plot, when we look at host associated, in particular, when we look at different mammalian guts. And actually, so we had that paper already a couple of years ago where we had looked at the dog gut microbiome, where we had argued that it's actually shares a lot with the human gut microbiome. But now in this case, we even have cats, and cats similarly seem to share a lot. So it does seem that, pets end up resembling us. We have this sort of visual joke from that. Yes. So, but then the next question is, okay, so some genes are shared, and which kind of genes? And some, not surprisingly, antimicrobial resistance genes, as well as mobile elements, are more likely to be present in multiple habitats compared to the to the baseline of all genes. And here actually we have, so in this plot I'm showing you all genes that are somehow classified as being antimicrobial resistant genes by RGI, but this includes different classes. And actually a lot of our ongoing work, and Svetlana is driving this, is also trying to understand which types of antimicrobial resistant genes are more likely to be present and to be able to cross these habitat boundaries. So maybe next time I can talk about those results, but today I just want to also briefly present the GMGC as a resource. So I'm going to switch here. So these are actually results where I previously queried the catalog with a sequence that happens to be a TET-A, a tetracycline resistant gene sequence. And so, I got a bunch of hits, and here we can see that the hits are across a variety of habitats. Actually, so most sequences would return hits restricted to 1 or 2 habitats, while here, not only do we have a lot of them, but here, so each one of these bars represents a hit. And you can see many of the bars are gray, corresponding to wastewater, but we also have a lot of these multicolored bars corresponding to these genes that are present in multiple locations. And we could then dig into where they are and click through. But in the interest of time, I'll let you do that at your leisure. So if you've been paying attention, you might have noticed that I've referred to to this as the GMGC version 1. And yes, even though that manuscript is still slowly working its way through the publication process, we are already starting to build the next version, which will be, even more samples, even more isolated genomes. And this is now a work in progress, but I just want to make the point that for us, this is a long-term project of trying to catalog and understand what is going on with the global microbiome. So to summarize just briefly, we use this approach of building gene catalogs where we take the genes from all the metagenomes that we find out there from these public resources, and then we sift through them to look for patterns of interest. In this case, we make the resource available to the whole community, but we are also internally very interested in this question of antimicrobial resistance gene and the cross habitat, genes that can cross these habitat barriers. I didn't mention this, but we do have tools as well. We have a tool called NGLS. There's actually a tool called GMGC Mapper that you can use to apply some of these resources on your own metagenomes, and feel free to email me or DM me on Twitter after that. And as I said, this is an ongoing project. Version 1 is done, even if unpublished, but version 2 will be even larger. So with that, I want to just briefly acknowledge that. So the Gene Catalog project was started. I was still a postdoc with Per Bork, and there's been many people working on this. This is not even an exhaustive list. And we're now working with on AMR with all of the people in the EMBARK consortium, but I'll maybe just single out Svetlana, who's been looking at this question of which kind of genes are and which kind of genes travel how and are present where. And finally, I want to thank the people who have funded me to work on this and funded the other people. And I want to thank you for your attention.
Thank you very much, Liz, for an excellent talk on how we work in EMBARK on quantifying genes at extremely large scales. So we have a few questions. I mean, feel free to keep bouncing questions in here. But I think I will make the format so that I will essentially pose questions to both of you here, because I think most of the questions we have gotten are pretty applicable to both of your talks. So let's start with one question from our own Etienne Ropé, who's also a PI in the EMBARK program, who I think was posted to Gerry, really, but I think it will be interesting to hear your take on this, Liz, after Gerry. So you mentioned that conventional antibiotic resistance databases mostly include resistance genes from culturable and pathogenic bacteria, and that hardly applies to samples where those bacteria are absent or subdominant, such as the gut microbiota or environmental samples. So what do you think about using specific databases to search for resistance genes in those kinds of samples? Let's start with you, Gerry.
Well, you're gonna miss stuff. Yeah, so that's what you have to realize. And I think that's one of the goals that we have in my group is to keep adding new genes that we've actually confirmed are bona fide resistance genes. I'll give you a good example. So the rifamycin resistance that you see in the clinic is almost exclusively due to point mutations in RPOB, the RNA polymerase. But in the environment, there's multiple mechanisms. Enzymatic inactivation, protection, obviously efflux is a big deal. And that's just one example. There's so many different ones. So for those, you really have to go out and hunt those genes down, use a functional approach. And then once you've got a gene, then you can use the databases and things like RGI and ResFinder that uses HMMs to be able to go in and look not just for the specific gene but for other ones that are close to it. And that's when we just have to keep adding to the databases so that we over time will be able to have a much more comprehensive approach. Right now, it's, as you point out, they're skewed, as you would expect, because that's where the data is, to everything that you can actually culture. But it really does, I'm a huge proponent of making sure that what we do, and we're adding these genes is in whenever possible, is to back that up with function, because you can get fooled where things you think are resistance genes, but they're not really resistance genes. So my plea is for making sure we don't pollute these databases going forward, but that we keep them as pristine as possible.
Luis, do you have anything to add there?
No, I mean, I agree with Etienne that this would be a good idea, and I'm actually happy to, because so we depend on databases like CARD, and so the better CARD gets, the better our results get. So yeah, so I agree with everything that was said.
So I have a continuation a little bit along the same lines, and this was a question that popped up in my own head regarding this. So there's a lot of different resistance gene database resources out there. So I mean, there's essentially two different ways I would like to ask this question. One is, why is that there is no single database that would benefit from all the individual inputs created from these different databases? That's how the question was posed in the chat. But I also sort of wonder, when we have this variety of different databases, which one should we choose? And I know that Gerry is biased because he has been part of the construction of CARD, but then maybe you can make a good argument for why we should be choosing CARD.
Well, when we built CARD, it was honestly, it was something that was ongoing in my lab for a long time. It was an internal resource, but one of the first sort of publicly available databases called ARDB came out just as we were pulling CARD together. And ARDB was, and at the worst, I mean, I don't want to cast aspersions to the people who built it, 10 years ago. Was not built by people who understand what resistance really is. It was a bioinformatic exercise, and it's still active. And the genes, there were 60,000 genes or something in it, and they weren't experimentally verified. So when we built CARD, we said every gene that we put in there has to be backed up by some peer-reviewed literature that says that this is a real resistance gene. So that's what CARD is. The RGI element of it, so CARD is the basis. I mean, it should be, the information in CARD is peer-reviewed literature. The RGI, which is a tool on CARD, takes that information and then uses, in this case, it's sort of, a blast approach to be able to figure out the strict and the perfect strict and loose strategies. And that's just one approach. ResFinder uses HMMs, and which can give you, I think, expand out a bit more the kind of diversities, kind of equivalent to our loose in some ways. So I think it just depends on what people came into it roughly around the same time and did. All I can say about CARD is that the data that it's based on, the core genes, are all from the peer-reviewed literature, and they should be unimpeachable. So I think it's a good data set to start with, no matter what your strategy is to do it. As to the very important question as to why don't we all get our acts together and just have one database, maybe that has all of these elements to it. The very simple answer to that is resources. None of these databases are funded. Everyone knows this in science. Anyone can build a database, no one can get it funded. So in the NCBI, as for an example, because it's an arm of the U.S. Government, they won't take in data from the outside the way that, so they have to build their own system. So we're victims in a field by the fact that this field in general is underfunded. Second, databases are around the world, no matter what you're in, are underfunded. And then we have these other barriers that are unfortunate. I can tell you that everyone in the field who makes these things gets along. There was an effort back about a couple of years ago to bring everyone together at a meeting in Italy, and there was a lot of agreement that we should do things together. but of course, all that agreement came with no money, and so CARD right now is run out of resources from our institute. There's no grant under CARD. It's just, we just keep it going as a labor of love. So great question. I wish I had a better answer.
Yeah, and I think, I mean, I agree with your view on why this is hard as well, that it's not just getting people to work together, it's also the resources around it and the long-term commitment and who is going to be behind that. So I fully agree on the problems with this. Luis, do you want to add something to this discussion?
No, I mean, you know, because you've been in some of these discussions, that this has been several hours in the internal discussion within the EMBARK consortium, exactly which approach to take. So we're probably not going to solve it in a few minutes here. I mean, to some extent, there is, you know, some of the decisions can be made a little bit rationally when you are trying to decide, you know, do you want to just, and RGI kind of has this approach internally. Are you just looking for, you know, these very good matches to things that are already known, or very good matches to things that are maybe clinically relevant, versus an approach where you are willing to accept a certain rate of false positives to broaden your search? Some of these decisions can be made a bit rationally, and some of the tools implicitly have chosen to be on the stricter side versus the, you know, versus a more this recall versus precision trade-off. But yes, it's not clear that there's a, you know, that there's a, you know, we've had hour-long discussions that it's not clear which approach, if there's an approach that would dominate, where everybody would say, okay, yeah, this is clearly better, so we should all do it this way. I don't, you know, maybe the field will eventually converge on something, but we're not there yet.
Yeah, I think, as you said, we have been having a lot of internal discussions about what database choices to make and things like that, and it is a complicated question, and as Gerry was alluding to here, I mean, that also depends on what you go into that task, what you're aiming to do. Moving topic a little bit, I think this might be slightly more aimed at you, Luis, so you get to start here at least. Do you perform any assays to describe the microbe composition in the samples in order to try to relate the antibiotic resistance genes to their carriers, or do you just capture your desired antibiotic resistance genes according to your objectives, is the question as the post.
So in our project, we mostly work with metagenomes that are publicly available. That we do. So there were, you know, people sequence them for whatever reason they had in mind, and so but we do have we do look at all of the genes, and we do have other measures. So for example, taxonomic abundances, which I didn't mention, that can be related back to the genes, and we can also, and this is not always possible, but if the gene happens to be, so say, in a larger contig, we can get their genomic context and try to start to dig in there. But generally, what we have is just a shotgun metagenome. So we try to exhaustively process it from the computational point of view, but we do not have parallel, or very rarely do we have some sort of parallel source of data.
So Gerry, when you work with your samples, do you relate them to taxonomic composition as well? I mean, you can see that you have done it in some cases there, but what's your general approach, if there is one?
Yeah, so as our objective right now is to use the Bates approach. When we do the Bates, for example, we don't just get that gene, but we get flanking regions as well. So our objective now is to pull that information into our analysis to be able to link it to us. And I noticed that this week, I think, or last week, Morton Summer actually published a really nice paper doing exactly this in Nature Communications. And so I think that's where the field is moving. So how do you add more? It's kind of a guilt by association in this case. It's so, you know, if you have this kind of gene, then you probably have this kind of association with a genus or a species even, and just by sequencing more and more, we start to make those correlations. And it also is really interesting because it should be able to help you start to predict. I think that's one of the punchlines of the paper, which I really love, which was, these kinds of mobile genetic elements and these kinds of resistance genes are associated together, and these kinds of mobile genetic elements can find themselves in all of these other genera. And so you can see, you can predict the trajectory of movement of genes through the environment. The more we get information about their taxonomic constraints of mobile elements. So it's a really nice beginning to where I think the field really should be going.
Yes. So actually, on the note here, you were talking about your bait capture approach there. So one thing that sort of struck my mind when you were talking about it is, I mean, to some extent I see that this could be almost a complementary approach to regular shotgun metagenomics, because I guess you get a much better picture of the incidence of different genes using this approach. But isn't sort of the abundance of the genes, the relative abundance, isn't that sort of obscured by the bait approach? Or how much bias do you get from the baits?
Yeah, great question. So we do see differences. So what the advantage is, we tile across the entire gene as much as possible. And so, you should see, if you've got relative abundance, it should be consistent across the whole gene, and not just a single region that says, well, this happens to sequence better than this other region. And so you can normalize according to that. But we've done a little bit of functional genomics in these samples, and the gene abundance that we're seeing, and it's not supposed to be a quantitative measure, but it does come out that you do see, a lot more beta lactamases and aminoglycoside resistance elements, which is sort of consistent with a functional genomic approach where we see a lot of those kinds of genes as well. So it kind of spills out of it, but I wouldn't want to use it as a direct measure of abundance. And the challenge with shotgun is to me, it just seems so random. So you may, if you have something that exists, if you have lots of something, then for sure you're going to see a lot, the chances are you're going to catch it. But if you make this the assumption that on average resistance genes are relatively rare in samples, then they're all relatively rare compared to hexokinase genes, right? Or ribosome components. And so the genes that we're interested in already represent a sort of thin group, no matter where you're looking. And so you're not going to really, I think, reliably get abundance out of shotgun, although I'd be happy to be wrong.
So I think we could have, it might be that we have a question from Svetlana. I will allow you to talk, Svetlana, and see if you will want to post a question, because you had a hand raised. That doesn't seem to be happening so much there.
She's on mute.
Yeah, but she is, I don't know, maybe this was just a mistaken raised hand. Okay, we are reaching 3 o'clock, so I think we have to cut the question session there. Thank you very much for attending, both to all in the audience, but also to Gerry, who got up really early to be here, and Luis, who has been staying up late to be here. I think there's a 12-hour time difference between you two. So thanks a lot. Before I end, I just want to again bring up the upcoming webinars here. As you can see, we have in 3 weeks' time, we have Heike Schmidt coming and talk about One Health surveillance. Then we have Debray Morris on the 9th of June, and on the last of June, Kimberly Klein. And then this will be followed in the fall by 6 talks, of which the 3 first talks have been decided, and that's Amy Pruden, Gavin Dantas, and Barry McMahon. So I hope to see you there. The registration links for the spring webinar is on our website, antimicrobialresistance.eu. And thank you very much for being here today. And I hope to see you again in 3 weeks.
Thank you, everyone.
Thanks. Take care. Bye now.